Hello guys, welcome to the first homework review session of season 7. We will be going over the benchmark quiz today. There were 24 questions, so I don't think it will take that long. And yeah, we will get started. Pom 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 pom. Let's see here. Can we all see the screen? All right. I will. Okay, this should be better. We will now go over the quiz. So, first question. When an ant dies in an ant colony, live ants will throw out the dead ant. If a live ant eggs is sprayed with chemicals extracted from dead ants, the colony will repeatedly throw out ant eggs. What is this behavior called? So, first we will go over what each of these means. Classical conditioning is what we read in Campbell. It's when you associate a neutral stimulus with another stimulus that is not neutral. It's like a dog associating the ring of the bell with the food, and that's why it salivates whenever it hears a bell ring. So it's not classical conditioning. Operant conditioning is based on positive reinforcement, positive punishment, or negative reinforcement, and negative punishment. It depends on reinforcing or encouraging or de-encouraging a behavior based on whether you want it or not. For example, if a mouse does something, you give it food. That's positive reinforcement. So in this case, there's no reinforcement going on. So it's surely not operant conditioning. Habituation is when something gets used to something. If you poke a baby, the baby, or if you put your, let's say, a finger in a small in the small hands of a baby, it will usually like clasp it. If you do it a couple times, it will stop clasping it. Exactly. If you poke a baby, it will usually clasp your finger. But if you do it a couple times, it will stop doing that and it gets habituated to it. So it's not habituation. The opposite of habituation is sensitization. That means the more you do it, the more specific, the more sensitized, or the more reactive the organism gets to that same stimuli. Taxis, there's different types of taxis. We have chemotaxis. Taxis is an attraction or repulsion from something. Positive chemotaxis is attraction to a particular chemical. Then we have haptotaxis, phototaxis, and so on. But in this case, the answer is fixed action pattern. The fixed action pattern is whenever the stimuli for the fixed action pattern is the chemicals that are characteristic of a dead ant. Whenever the ants smell that chemical, it will throw out whichever ant has it. The link should be in the chat. So yeah, the first answer is fixed action pattern. I believe this should not have been a problem for anyone. Are we good? Okay. This is a Bayes problem. I was surprised we decided to have Bayes as the second question, but yeah. So we will first go through this together. We know this is AA. And this is you're pretty sure at this point that this is an autosomal recessive. Bayes is a probability technique, which we will go over right now. This is small a, small a. This has to be big A, small a, big A, small a. Because both of his parents are fine. That means they have at least one big A. And you see that there's one of their offspring has homozygous recessive. That means both of the parents also must have one small a, small a. So yeah, you got that much. And now what is this offspring? You know that he is unaffected by the disease. So there's a 1/3 chance he's AA. There's a 2/3 chance he's AA. Are we good so far? Do we realize why it's 1/3 and 2/3? Because you know that it's not small a, small a. Because if it was small a, it would be colored, filled in, and the person would be affected. 2/2 would be affected. And the question tells you that this is big A, small a. We know this much. And we know the offspring is unaffected. We don't know what the genotype is. It could be AA, it could be AA, it could be whatever. So you want to find the probability that 2/2 carries the disease. So the probability that 2/2 is AA. Carrying the disease means it's heterozygous. Probability that 2/2 is AA given that 3-1 is unaffected. This is a symbol of probability. So if you have like P, A, B, it means probability of A given B happens. So intuitively, this is how it makes sense. It's like you're updating the probability. If you didn't have this child, you could confidently say that there's a 1/3 chance it's homozygous dominant and there's a 2/3 chance it's heterozygous. However, now that you know that the first offspring that it had was not affected, that means there's a higher chance, kind of, that it's this rather than this. A slightly higher chance. So you are updating the previous probability. You can also, if you have taken AP stats, you have probably encountered tree diagrams. This is exactly the condition of a tree diagram, except in this case, this is exactly a tree diagram. And you can do it by drawing a tree diagram. However, that takes far too long. Yeah, drawing a tree diagram is tedious, to say the least. It's not tedious, but it takes long, and that's what you don't want to do at USABO. So at USABO, Bayes has a formula. P given A, P of probability of A given B is the probability of B given A times probability of A over probability of B. Now, in this case, we already defined A and B. A is the probability that this person is heterozygous. B is the probability that 3-1, the offspring, is unaffected. So now we plug numbers in. So probability of B given A. So probability of 3-1 being unaffected if the dad was capital A, small a. So if you have a cross of AA and AA, the probability of getting an unaffected offspring is 3/4. Do we agree on this so far? Okay. So PBA is 3/4. Then we look at probability of A. So probability that this mom is heterozygous. We already said that that probability is 2/3. Yes, exactly. So we have 2/3 right over here. Then the probability of B. Probability of B is the probability that this offspring is unaffected. You have to account for two probabilities here. First, the probability that this guy is homozygous dominant, which is 1/3. If that guy is homozygous dominant, then the probability that this offspring is not affected, it's 1, because the offspring will always inherit a dominant allele from the dad. And if the dad is heterozygous, which is 2/3 chance, we previously said that, then there's a 3/4 probability that he will be unaffected because of what we said here. If you look at it, 2/3 times 3/4 over 2/3 times 3/4 plus 1/3 times 1. This is exactly how a tree diagram formula looks like if you have taken AP stats. But if you do this out, you'll get half over 1/3 plus half. So this will become half over 5/6, 3/6 over 5/6, which is 3/5. And 3/5, I believe, was the correct answer. Are we good with this? If you have problem with Bayes, yes, the mother was already said in the question to be AA. If you read the question, it says that the mother is a carrier. Carrier is heterozygous. Could I check Q&A? Please just put your questions in the chat, please. Q&A is difficult for me to check. I will check Q&A at the end of the class, but I know that the writing is... I know what the writing means, but I don't know what is in this context. Okay. I will explain it again because I feel Bayes is important. I will explain this and I'll probably explain the Hamilton's one too. So, how do I explain this again? I will erase everything and rewrite and do this again. So, you know that this son is small a, small a. You know that because of autosomal recessive. So, their parents have to be big A, small a, big A, small a. As such, this person has a 1/3 chance of being big A, and 2/3 chance of being big A, small a. The question already tells you that 2-3 is a carrier. So this is a small a, this is a heterozygous. The offspring is unaffected. I already explained what Bayes is to you. So I will show you what the probability. The probability is you have 2, 2 is A, given 3, 1 is AA or AA. And the probability of that is P of B. Probability of B given A. Probability that the child is unaffected given that the dad is a heterozygous carrier. That's going to be 3/4. The probability that the dad is a heterozygous carrier, that's 2/3. We can copy this again, 3/4 and 2/3. Plus, the probability that dad is homozygous dominant, and in that case, there's a 1 chance that the child is unaffected because the child will be unaffected. You know that. In this case, you just cancel this out. Fuck, I canceled the wrong thing. But half over half plus 1/3, and this will give you 3/5. If you want, we can talk about. Kevin will cover Bayes in genetics, and Bayes will take as Deep said, Bayes takes a while to understand and do fast on an exam. So it takes a practice. Oh, God. This is a plant root. So yeah, gravity is sensed in the root cap. Yeah. Gravity, no, it's not one. One is like a tip of the mucilage. Gravity is sensed in the root cap. 3 is like the apical root meristem. So it's 2, like in the root cap. Campbell just says it. If you want to ask me why, it is because of statoliths to some extent. The mechanism by which it occurs is that the auxin falls like here. So if the root is like this, so in this case the root is like bent and it wants to like it's bent. It's not like straight as it should be. So in this case, all the auxins fall on this side of the ground. Auxins cause calciums and so on, and there's a signal transduction pathway, but you should remember too much auxin inhibits growth. Little auxin promotes growth. Too much inhibits growth. Because of too much growth is inhibited here. If growth is inhibited on this side, then this side will grow longer and longer. And slowly over time, you will see the root corrects for the fact that it was bent. That's the mechanism. And that's what you will see in Campbell Bio, chapter 37. Actually, 39, 38, I think. 8 or 9. I get it's whatever plant chapter there is. Just read Campbell Bio. Do not ask me why or how. If you want, 39, thank you, Jacob. It's a plant hormone chapter, I think. Our plant responses to internal and external stimuli. Okay, so what's the most frequent origin of Down syndrome? Down syndrome, as you know, is trisomy 21. It is because of non-disjunction of chromosomes in meiosis. So polyploidy is not polyploidy. Aneuploidy is usually caused by non-disjunction. You should just know this. Down syndrome can also be caused by Robertsonian translocation. I don't want to go into it. Kevin will talk about it when he discusses his stuff in genetics, and that's not my business, and people will get confused if I do it. So yeah, I will keep going. So acetylcholine. For those of you who don't know, wait, what? Evan, what did we do? I think that's the one that dependent on that if you get Down syndrome because of translocation, I think that's what happens. Maybe, I don't know. I don't exactly know. I just said stuff. Why are you doing problems unrelated to the chapter we are on though? Whoever asked this, there's a benchmark quiz. It's a benchmark quiz. You're supposed to have every topic on this. But I guess, yeah. So Let me tell you about a neuron. These are the beautiful dendrites of a neuron. This is a beautiful cell body. This is the beautiful axon hillock. God, I'm terrible at drawing. Bloop, bloop, bloop. These are the axon terminals. Now you zoom in. You see at the axon terminals, you have these vesicles. You have this calcium ion influx. So when the calcium ion goes in, these are like primed. These vesicles are Mbach calls them primed. They're primed to bind and release the neurotransmitters. I think the calcium ion binds to synaptobrevin. I don't think it's syntaxin, right? I think it's synaptobrevin. Yes, synaptobrevin and synaptotagmin. Yes. Also, by the way, these are the proteins that are involved in T and V snares. T and V snares are proteins on vesicles and target membranes. Yes, SNAP25 is also there. That causes fusion of vesicles with their target membrane. You can look at MBOK chapter 11. They explain it really well. But yeah, in this case, these vesicles are filled with acetylcholine. And of course, if you go a little bit far away, there's another dendrite. That dendrite has receptors for acetylcholine. In this case, I believe it is nicotinic acetylcholine receptor. But yeah, we don't need to know what that is yet. That's session 2. These are ACH receptors. Now, the acetylcholine that's released into the synaptic cleft, they bind to these ACH receptors. When they bind to these ACH receptors, if it's a nicotinic acetylcholine receptor, it causes the opening of ion channels. If it's a muscarinic, then it activates GPCRs. But you don't need to know that. All you need to know is that it activates the postsynaptic neuron. Now, if there is no cholinesterase is in this synaptic cleft area. Cholinesterase breaks down acetylcholine into acetyl plus choline. And then this gets taken up by surrounding astrocytes in the CNS, or this can get like taken up by the presynaptic neuron, and the presynaptic neuron can metabolize it back into the acetylcholine. So yeah, if you don't have the cholinesterase, the acetylcholine will just be there, and the postsynaptic neurons would fire in a rapid, uncontrolled fashion. Are we good? I hope so. Okay. Just remember, this is a definition from Campbell Bio. Yeah. Just also remember, this is also in ecology from Campbell Bio. Yeah. Just also remember, if you look at this, it's obviously wrong. Biomass ingested by consumers over available biomass. This is exploitation efficiency. I think if someone thinks it's something else, correct, it's incorrect. Yeah, I thought the same, but then I saw these two and I was like, yeah, this has to be this. Exploitation efficiency is what this is talking about. Production efficiency is the amount produced by like the herbivores, plants, and stuff over total amount of energy given by the sun or something like that. What is the effective population size formula derived from? Very good question. I believe it is derived from like there is this something called Waterson's coefficient. I know it is somehow related to effective population size. The relationship, I don't know yet, but I will cover that in a skill building at some point. This is PopGen, so you will wait until we cover it. Are we good so far? Yes, wait, this is not an easy formula to actually derive, and this is not the actual formula either. Effective population size is much more complicated than this. And like, this is not a formula. If you guys know Eric Lander is like a very famous person who studies the effective population size of humanity itself. And like, this is a complicated topic. So like, yeah. I don't know, bro. I don't really know whether he's racist or not. He's a professor at MIT. All right, Gavin, minus 1 million Baobucks. You cannot be saying that shit in class. Okay, we will start. Just kidding. You won't get minus 1 million. You get minus 10. It's okay. Okay, so this is a tri-hybrid cross, and we're doing linkage mapping. It's not a tri-hybrid cross, I guess, but whatever. We have this. These are the parental types. The most common ones are the parental types. The least common ones are the double crossover, DCO. So if you compare the double crossover with the parental, whichever one, there's one gene that's wrong. That gene is in the middle. So you see this. A and B are fine. A and B are fine. C is not fine. C is capital, C is small. Then you look at this. A and B are fine with A and B here, but C is not fine. So A, C, and B. Yes. And you have to find distance between A and C. That's 5 plus 8 because both A and C have to be capital or both A and C has to be small in the parental. This is fine. These are the parentals. This is not fine. This is not fine. So 81 plus 85 plus 13. That's 166 plus 13, so 179 over 1000. 0.179 centimorgans. Are we good? Can we explain in general how to find distance? First, you need to find the gene in the middle. In order to do that, you have to find the double crossover. The double crossover is the group with the lowest amount of offspring. You look at the double crossover and you compare it with the parental. You will see that there's one gene on the double crossover that's messed up, and the two other genes are the same as the parental. That one gene that's messed up is the one that's in the middle. And from that, you can find the distance between each of them. So if you want to find the distance between, let's say, C and B instead, you look at how C and B are in the parental. C and B are both capital or C and B are both small. So any case where C and B are not both capital or aren't both small, those are the recombinant ones. So this is recombinant, this is recombinant, this is recombinant, this is recombinant, but these are not recombinants because you see both B and C are small as in the parental. But you should not skip this. This is a simple enough question once you know how to do it. So phenograms. This is just a fact that you need to know what phenograms look for. Phenograms, if you look at it's phenotypes probably. So physical similarities, yes. Physiological similarities, yeah, both are technically the same. And ecological niches, if you know the physical similarities and the physiological similarities, ecological niches is also like the phenotype of an organism, kind of. If you really, really think hard about it, the phenotype of an organism determines its niche. So technically, if you group an organism by its niche, then you get the phenogram. How did I select this? Anyways, so O blood group is known as an universal donor. So O blood group, the red blood cell itself has nothing on its surface. No A or no B. So no A, no B, isoagglutinin. However, its plasma, bloop, 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 has both anti-A and anti-B agglutinin. So O is the best donor and O is the worst acceptor because regardless of whatever blood you put into O, unless it's O itself or unless it's a Bombay phenotype or whatever, it will reject it. AB is the worst donor because A has both the antigens or the isoagglutinin. However, AB is the best acceptor because regardless of whatever you put, it will accept it. And this is the correct type of plasma, has both anti-A and anti-B agglutinins, as I said. Type of red blood cells contain isoagglutinin A and B. No, they do not. That's why they're good donors, because they don't contain either. Individuals with type O blood can be transfused with blood that is genotypically AB. No, they have both anti-A and anti-B. You will get cooked. There is genetic advantage to typing type O blood. No. Having type AB blood means you can accept anything. But having type O blood, that means you can give stuff to others. Giving your own blood to others does not make you evolutionarily superior because it doesn't help you become stronger yourself. So, no. Type O blood is determined by the presence of three alleles. That's just simply not true. Oh God, again, plants. Why? 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 Anyways. No, isoagglutinin and anti-agglutinin are not the same. Isoagglutinin are on the red blood cell surface, and anti-agglutinin is like in the plasma against that. Isoagglutinin are like, if you have isoagglutinin for type A blood, then you have type A blood. If you have isoagglutinin for type A blood, that means if you put in type A blood, you will get cooked because the anti-agglutinin will attack it and cause agglutination or clotting. Yeah. The answer for this is supposed to be less total root surface area and thicker layer of suberin. Now, I will tell you my thoughts. Higher concentration of ethanol in water doesn't make sense because here we are not doing ethanol fermentation like animals do. Higher root density doesn't really make sense because it's already waterlogged, so it's already cooked. Higher concentration of hydrogen sulfide doesn't make sense either because you are not doing anaerobic respiration like hydrothermal vents. Thicker layer of suberin can sometimes prevent stuff from getting into the roots that may be toxic or shit. So like it's fine. Less total surface area. Oh, sorry. I'm sorry, Evan. I will be more conscious of my language. Anyways, less total root surface area makes sure that you are not. I guess, bro, you can take away minus 67 Valbucks from me. Less total root surface area is useful when it's already waterlogged. If it's already waterlogged, then there's no point in spreading your roots. A plant spreads its roots and grows more when it's in a nutrient-rich area. If it's not in a nutrient-rich area, if it's like in a nutrient-deficient area, then it like kind of lets go of the roots that are extra. Because remember, every piece of root gets energy from the leaves. If there are tons of roots, that means each piece of root gets that means the energy produced by the leaf is distributed among all of those. That's inefficient if the root isn't doing something. Now, for this question, I'm sure that the answer key says dorsalized features. I'm here to tell you that's not correct. It's a normal embryo. Animals can do fermentation, yes. Is there not fermentation though? There's no ethanol fermentation in plants, as far as I know. There is? Alright then. This question has problems with it in terms of answer key. It's plants question, so you should not ask me to correct this. However, there was a problem with this question, and I can correct it. So I will tell you guys about animal hemisphere and vegetal hemisphere. The animal hemisphere has no yolk. The vegetal is filled with yolk. Bloop, 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 bloop. Typically, a sperm enters. Alright, guys, what's wrong with the bloop? Why are we so mad about this, bro? Mm. Alright, we will be taking a 2-minute break for me to drink water. No, it is water. Never mind. I should not say what I was about to say. Okay, so Xenopus laevis is a type of frog. The sperm usually enters at the animal hemisphere. I will also let you know that the cortex of the animal hemisphere is gray. That's because the animal hemisphere has a lot of nuclei in it. If you have gray cytoplasm or like gray cortex, then it absorbs the warmth of the sunlight. And it kind of protects it also from the sunlight because yes, it just does. That's how like it also like it gives warmth to like the nuclei in the animal hemisphere and also protects it from like the UV rays and so on. So that's why it's beneficial to have a gray cytoplasm. Here comes the sperm, it goes into the animal hemisphere. Then we have a cortical rotation. towards the point of sperm entry. And at the end, we will see this, and there will be this area that's exposed. The cytosol is gray, right? So this becomes a gray crescent. The nucleus center forms right under the gray crescent. The nucleus center is formed by the dislocation of disheveled GBP. From here to here when the cortex rotates. Disheveled GBP is a part of the Wnt beta-catenin pathway. So in the Wnt beta-catenin pathway, you have Wnt, which binds to a cell surface receptor. It activates this protein called disheveled. Disheveled inhibits this complex of actin and GSK3, among other proteins. And they actively inhibit beta-catenin. And beta-catenin causes cell proliferation. So by putting disheveled GBP, you are inhibiting this, so beta-catenin is active and it can cause cell proliferation. The nucleus center later on induces the Spemann-Mangle center at the gray crescent, but that's a story for another time. But yes, at the frog embryo, the first division that happens is like true, so you'll get two halves that both have the nucleus center right over here. And then the next division happens like right into the page. Like right into the page here. In that case, you will get two that don't have any nucleus center and two that will have half of a nucleus center. These two will become normal. These two will become ventralized. Why is the animal-exposed cytoplasm gray? I don't know why it's gray, but it's evolutionarily, it's beneficial for it to be gray so that it can protect it from sunlight. Because the animal pole has more of the nuclei. If you remember, the vegetal pole is filled with yolk. Most of the divisions are like mesolecytal. We will cover this when we get to anatomy and physiology. So we will get a normal embryo, not an embryo with dorsalized feature. It's very difficult to get an embryo with dorsalized feature. You have to do external interventions usually to do that. So we will get a normal embryo. Yeah, so this is an easy question. First, you need to get the sequence from the Sanger sequencing shell. You start from the smallest one. So you start from A. A, T, A, T, T, G. T, C, G, T, A, G. 3 prime. And then you write the template from it. So G is C, T, A, C, G, A, C, A, T, A, T. So 5 prime is C, T, A, C, G. C, T, A, C, G, so it's A. This should not be difficult to do. You have to realize that this is the template strand. This is the template strand, 5 prime to 3 prime. Then you're making another template strand from 5 prime to 3 prime. And that's of varying lengths. And those are the ones that are on the Sanger. So you have to get whatever is on the Sanger from the smallest to the biggest. This is the smallest. The smallest ones float the furthest down. These are the wells. So you make it from the smallest to the biggest, and then you find the complement of it. I hope that makes sense. Oh, this is finally a nice question. Wow. So I will tell you guys how the eyes look. Bloop, 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 bloop. Yes, so damage to the optic chiasm, this is the optic chiasm. That means you don't get the left field of the left eye and the right field of the right eye. You will only get the center. So it would not result in complete blindness. False. Damage to the optic chiasm will still allow visual input from each eye to reach the occipital lobe on the same side of the brain. If you disrupt this, then this wouldn't get through, this wouldn't get through. However, this would get through, this would get through. And this is in the left eye, it goes to the left occipital lobe. This is in the right eye, and it goes to the right occipital lobe. So this is correct. Damage to the left occipital lobe will impair vision only in the right eye. That is false. Damage to the left occipital lobe, this is the left occipital lobe, it will impair vision of the right visual field in the left and the right eye. Does that make sense? Because you see this and this, these are what's in the left occipital lobe. It will inhibit the right field of vision in both eyes. Damage to the right occipital lobe will impair vision from the right half of the retina of each eye. That is completely true because if you take away the right occipital lobe, then this is the right, then this looks at the right, this looks at the right, this is the right and the right. So damage to the right occipital lobe would impair vision from the right half of the retina of each eye. This looking to the left. You can see that this looks upon the left, but however, this is on the right side of the retina. So this is true. Does that make sense? Yes. So the first one is false because it would only cause central view, not peripheral view. Second is true because the outside peripheral ones would be active. Damage to the left occipital lobe would impair vision in the right visual field of the right and the left eye. And damage to the right occipital lobe would impair vision in the left visual fields of both eye, but in the right half of the retina of each eye. And this is just hydrophobic interaction because that's how proteins are. Most of the interactions are hydrophobic interactions. So even though they're much weaker, since there's more of them, they're the most important. Goiter may be caused due to iodine insufficiency. TRH, thyrotropin releasing hormone, causes the release of TSH. TSH causes the release of T3 and T4. T3 is the active form. This is released by hypothalamus. This is released by anterior pituitary or adenohypophyses. It's formed to the Rathke's pouch and all that, but we will cover that at a later time. This is released by the thyroid gland. Bloop, bloop. So, high levels of tyrotropin releasing hormone will act on the thyroid gland and make it get bigger because they're tropic hormones. Tropic hormones, when it acts on the gland, makes it bigger. High levels of thyroid stimulating hormone would also do the same. High levels of thyroxine and triiodothyronine would inhibit TRH and TSH release, and that would cause the opposite. That would cause atrophy of the thyroid gland because there's no THAs or TRH. There's no TRH to stimulate TSH and there's no THAs to stimulate the thyroid gland. And because of it, the thyroid gland will atrophy and it will not be able to release hormones later on. So this is something that would not cause a goiter. If you look at this, it will be very obvious to you that this is autosomal dominant. You have equal proportion of men and women, so it's not sex linked, and you can see this is why did I say recessive? This is dominant, yes. Yes, I circled wrong one. But this is dominant because as you see, it doesn't skip a generation. Oh, goodness gracious. From my meager understanding of plant bio, this is early wood. And this is late wood. Yes? What is it about, Udeep? Young versus new. This would be younger then. I think the right will still be younger. Because, oh, I got you. Everything kind of makes sense now. So, there's two types of typically xylems and stuff. One type has bigger like this. This is for transport. This, when you have like smaller of these like tracheas and whatever, this is usually for support. So, usually the support ones, like you have heard of heartwood, right? Heartwood is in the center, and heartwood is supportive. So this is in the center, and this is away from the center. So you go to the right. Did that make some sense? Because xylem can have two purposes. If xylem is big tracheids, that means it does transport. If it's a small trachea, that means it does support. Support is usually in the center. And right, in this case, the big ones are on the right. That means on the right, you have the younger ones because the center are away from the center. And if you want to get out of the tree, you want to get away from the center. So you go to the right. Are we good? Why can't I just move up or down? Because a tree may be like a oak tree and infinitely long, or a redwood tree. The tree fell down. We guess, bro. We guess. For this question, the answer is just B and E. Each of them absorb the wavelength at their own characteristic like frequency. So that's just how it is. Oh, Lord goodness gracious. Anyways, okay. This is a Hamilton's problem. But I want to get to the last two questions first because these are easier and then we will do the Hamiltons together. So in this case, we have a fish. The fish was breathing fine with the water, and then some idiot decides to grab it out of the water and check how long it can breathe and its oxygen saturations. So, total blood saturation by oxygen. How do we find that? That's a bit hard to find. So, first, we will look at oxygen supply through gills. That's more obvious, right? Oxygen supply through gills should fall immediately and be very, very low, very, very near zero. So, it's fair to say that this is oxygen supply through gills. We also know total blood saturation by oxygen should also decrease. So this is A. One of the main things you have to distinguish is which one is A and which one is B. In this case, the one that stays near zero is most probably B because on the land there's no use of gills. And if you look at that, you only see one option with one A and two B, and that's this one. And that's the only one that makes sense. Yeah. So what happens during meiosis and mitosis? In meiosis, it increases once, then it decreases during telophase 1, and then it decreases again, which is 2. And this is mitosis because the symbols, so 2 and 1. Did we get this so far? Now we will do the obnoxious Hamilton's one because I hate Hamilton. This is not a hard math, but I hate Hamilton's regardless. So you have a grandma, uncle, and first cousin. I hate this equation. So I don't use this equation. I use a different equation. I use with help must be more than without help. So I will teach you how to do this. You have to calculate gene by core. Or you have to calculate number by core. Core is coefficient of relatedness. So let's draw a pedigree for this. You need a grandmother, uncle, and first cousin. Bloop, 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 bloop. 67x, exactly. So the grandmother is this. So this is the kid who is supposed to like save whoever. His grandmother is this. So in order to find coefficient of relatedness, you draw lines. Each lines represent half. One line is a half. So how can you go back to the grandmother? 1, 2. This is the only way you can go back to the grandma. So there's 2 lines, so it's 1/4. Then you have to go back to the uncle. So there's how many ways can you go back to the uncle? 1. So there's one way, and that's 1 times 2 to the 3 because there are 3 lines. Is there another way? Yes. 1, 2, 3. So plus, and this is 1/4. So plus 1/4. Then you have to go to the first cousin. 1, actually, this is becoming a mess. I will make the first cousin like thingy bigger. 1, 2, 3, 4. Or 1, 2, 3, 4. 2, 3, 4. 4. So you have 1 times 2 to the 4 plus 1 times 2 to the 1 over 2 to the 4. That's 1/8. So this is the total coefficient of relatedness. 1/4 plus 1/4 plus 1/8. That's 2/4 plus 1/8. That's 5/8. Wow, math is hard. Do we agree so far? Could you just do it intuitively? The intuition is that this is the probability. The reason one line is half is because you have two sets of genes. The probability that you are taking one set of gene and it's the one from your mom is half, because there's a half chance it's from your dad and there's a half chance it's from your mom. So each line is a half, and you make lines to connect two descendants together. If there are multiple ways you can connect them together, then you add those multiple ways. That's the intuition. If you read Griffiths, it'll teach you what identical by descent is, but that's more related to coefficient of inbreeding than coefficient of relatedness. But yeah, that is supposedly the intuition. So I guess with help, 5/8 is whatever the coefficient of relatedness is. With help, the probability, they have a 25% of chance without your help. But with help, they have a 75% chance. So 3/4. And if you help them, your gene itself is 1. And there's a probability P that you survive. P is a probability that you survive. Remember that very well. Without help, 5/8, and there's a 1/4 chance of survival. So 1/4 plus 1, because you will survive in that case. You want to say with help is higher than without help. That's the only way the guy is going to go and help his family. So with help is going to be 15 over 32 plus 1. That must be more than without help, which is 5 over 32 plus 1, which is 37, 32 plus P is more than 15 over 32. So P must be more than 37 over 32 minus 15 over 32. That's P must be more than 22 over 32. Do we agree so far? No, what do you mean no, bro, Nicole? Do we agree? What? Okay, all right. Do we understand how we got here? We write two equations. One is for with help. If we help them, then the chance that 5/8 is a coefficient of relatedness, which we have here, and the chances that they will survive is 75%, so 3/4. And if you help them, there's a chance that you will survive. Your gene itself is 1, and the chance you will survive is P. If you don't help them, then there's a 100% chance you will survive, of course. But if your family surviving is 25%. And you say with help must be more than without help. And you find P given that. And in this question, it asks you, what is the maximum likelihood of drowning? So P is a likelihood of surviving. You want to find a likelihood of drowning. So you do 1 minus P. In that case, P must be more than or less than 10 over 32. If you put 10 over 32 in your calculator, I will actually put 10 over 32 in my calculator. It says 0.3125, so it has to be this. And hopefully that is the answer. And we went through the entire test. We are in fact done. Do we have any other question of pressing matters? Are we good? Wow, thank you so much. What can I say? Explain the neurons. Loop neuron releases acetylcholine. Loop, another neuron. It binds to the acetylcholine. If there's no choline esterase, then the acetylcholine will just remain in the synaptic cleft. And okay, just think about this is a neuron. it releases acetylcholine. This is a neuron. It has receptors for acetylcholine. Now, if there's no cholinesterase, then the acetylcholine will just remain in the synaptic cleft between the two neurons' membranes, and it will just keep activating the acetylcholine receptors. When it keeps activating the receptors, it will cause like excessive signaling. So if you don't have cholinesterase, it will cause excessive signaling of the postsynaptic neuron. Yes, read MBOG. MBOG is a great book. As for mitosis and meiosis, remember during S phase of mitosis and meiosis, things double. Then during anaphase of mitosis, things become this. However, meiosis during S phase, things double. Then during anaphase 1 and telophase 1, things half. And then anaphase 2 and telophase 2, things become this. So 2n to 1n. And we know meiosis from gametes. And mitosis 2n to 2n because mitosis just duplicates the cells. That's developmental. For developmental biology or embryology, you want to read Gilbert's. Gilbert's is a very cool book. That's where I got this information about new Coop centers from. Yes, this is very cool. Or like, I'm not screen sharing. I forget, bro. I forgot. But anyways, it's fine. Wait, I can't. Wait, I don't have his though, bro. Go and ping Joseph on Discord. He will give it to you. Yeah, Joseph has the histo. Don't ask me for histo. I can't help you with it. But yes. Do you want the answer for the phenogram species question? Because the phenogram species question, pheno means a phenotype. Yes, pheno means phenotype. And phenotype is something that's externally observable. That definitely includes physical and physiological traits. However, remember that the ecological niche is like the niche or it's like the role that an organism plays in an ecosystem. And that is very determined by its physical and physiological traits. So, if you group something by its ecological niche, that tells you stuff about its physical and physiological similarities. So that's why it's all three of those. However, remember physiological and physical similarities does not imply evolutionary relationships because of convergent evolution, and you can have analogous structures. But yes, that will be class for today. How is beta-catenin? Beta-catenin acts as a transcription factor. It binds to TCF and left family of transcription factors and activates them. And when it does, it causes the transcription of cyclin D and other genes. Cyclin D is very important because cyclin D is one that binds to P21, P27, and P57. And after binding to those, it can activate cyclin E. And cyclin E is what causes transfer or like transition across the R point or restriction point. Analogous is for cyclin E CDK2, yes. And cyclin D binds to CDK4 and CDK6. And yes. What did I want to say? Yes. Analogous is convergent. Homologous is ancestry. Orthologous is two different species, speciation. Paralogous is duplication in one species. Orthologous is like the beta globin family in like dogs and us. Yes, you can leave, Aaron. And paralogous is like the different families of beta globin, like alpha globin, beta globin, and so on, that we formed in our own genome by duplication. Anyways, that will be everything. Thank you so much, and I will leave now. If you have questions, please ask in the Discord.